Which is a nitrogenous base present only in RNA, not in DNA, that is uracil. Remember, the alpha and the D forms can be written based upon the general structure of the glucose. If the amino acids are synthesized in our body, it's not necessarily taken through the diet. Such amino acids are called as non-essential amino acids. Remember, to form a peptide bonds, we must have two amino acids. So when two amino acids are formed, one peptide bonds are formed. Hello my dear students, a warm welcome you all. Myself is Purnima, lecturer in Department of Chemistry at Vidyashram Pre-University College, Temple of Excellence, Mysuru. My dear students, today we are going to start with the revision class for the Unit 14 Biomolecules. So let us see what we are going to discuss with this unit. In this unit, we are going to discuss more about carbohydrates, proteins, amino acids, then enzymes and also with the vitamins. And in addition to that, we are also going to discuss with the nucleic acids. So as we all know that carbohydrates are also biomolecules which play a very important role in a human body, correct? So in this unit, the structure and function of some of the biomolecules will be discussed, and the structure and function of the biomolecules inside the living body studied in the biochemistry, and the living systems are made up of various complex biomolecules such as carbohydrates, proteins, enzymes, lipids and vitamins and hormones, and the nucleic acid, which are also compounds which are helpful for the storage and exchange of energy materials such as ATP, etc. Remember, in the first part of this unit, we are going to discuss with the carbohydrates, proteins, amino acids, vitamins, then with the hormones. And in addition to this, in human body, we know that there is a transfer of characters from one generation to another generation. That is mainly because of the hereditary material called as nucleic acid. This nucleic acid will transfer some of the characteristics from ancestors from one parents to other generations, correct? So that is also we are going to understand in this unit, which are the very important biomolecules which also kept for the studies. From this unit, we can expect 2 5-mark question. Okay, and remember, again the 5-mark question can be directly divided into 2 plus 3 or 3 plus 2 or 2 plus 2 plus 1. So for the sake, I'll discuss both 1 mark, 2 mark as well as with the 3-mark question which has been raised many times from this unit. So the 1-mark question goes like this. What are the expected products of hydrolysis of sucrose? We all know that sucrose molecular formula is C12H22O11. Upon hydrolysis, in presence of a suitable acid, it gives C6H12O6 plus C6H12O6. So this is your glucose and this is your fructose. Okay, glucose and fructose. Name the amino acid which are containing sulfur. As I said, there are 20 different types of amino acids will be there. So, whose structure along with their types, especially with the neutral amino acid, with their acidic amino acid as well as the basic amino acid, you must know that at least with the structure as well as the name. So, in that 20 amino acid, you must know that what is the exceptional cases also with the compound. So, the sulfur containing amino acids are cysteine and methanoine. Okay. Next question is, what is Zwitter ion? We know that when we take the amino acid, when we dissolve it in water, the carboxylic groups of amino acid will leave the hydrogen. That hydrogen will go and attack for the basic groups of the amino acid, that is NH2, and forms NH3. The presence of both positive and a negative ion, that is a dipolar ion, if it is formed an aqueous solution of amino acid, which we call it as what? Zwitter ion. I can show with the reaction now. So we write the amino acid general formula CHCOOH NH2, correct? So this is your basic group, this is your acidic group, correct? And is your R is a variable group. Now if I dissolve this salts of amino acid in water, As I said, the acidic group will leave a hydrogen. This hydrogen will be accepted by this basic group and forms what? NH3 plus CH, COO minus and R. The presence of both this positive ion and the negative ion, if it is formed in an aqueous solution, which we call it as what? Zwitter ion. So it can be asked with the reaction for the 2 marks also. Next, what is isoelectric point or the isoelectric pH? The pH at which amino acid acts as a Zwitter ion in aqueous solution, which is said to be what? Isoelectric pH or the isoelectric point. Next, what is a peptide bond? Peptide bond, imagine you have one amino acid, okay? So there is another amino acid. These two amino acids are linked together by CONH bond, okay? So the CONH bonds are formed by elimination of water molecule, okay? This is known as what? Peptide bond. So peptide bond is a amide bond which is formed between two amino acid units with the peptides and a protein. Next, which is a nitrogenous base present only in DNA, not in RNA. So we know that in DNA, what are the bases will be there? The bases are adenine, guanine, cytosine and thymine. Whereas in the RNA, we have a nitrogenous basis called adenine, guanine, cytosine and uracil. So the common thing is, in both DNA and RNA, we can see adenine, guanine and cytosine will be there. But thymine will be found only in the DNA and uracil will be found only in RNA. Clear? Next, which is a nitrogenous base present only in RNA, not in DNA, that is uracil. Okay. Next, name the hormone which control the carbohydrate metabolism. Example, insulin. Okay. Next, how many peptide bonds are present in a pentapeptide? Remember, to form a peptide bonds, we must have a 2 amino acids. So when 2 amino acids are formed, 1 peptide bonds are formed. Similarly, how many peptide bonds are present in pentapeptide? Pentapeptide in the sense, there are 5 amino acids will be there. When they link to one another, they forms a 4 peptide bond. Next, name the water insoluble component of starch. So amylopectin is water insoluble content in the starch. Next, we'll go with the 2-mark question. Write the Hawa structure of glucose. So remember, the glucose structure will be consist of 6 carbon atom, correct? 3, 4, 5 and 6. And we know that it has a 1 carbonyl group, and this carbonyl group is nothing but is your aldehyde group, and we have OH. Okay, there are 5 OH will be there. H, OH and OH, correct? So you have to fill with the H. Okay, and we know that the oxygen atoms in the glucose, can be attached to this carbon atom, thereby shifting of bond will be taken place and forms what? Structure of glucose with a alpha and a D form. Remember, the alpha and the D forms can be written based upon the general structure of the glucose. Okay, so in this structure you can see that after writing that alpha form, CH2OH, and these carbon atoms are linked by oxygen. Then we have OH, then we have H, then OH, then we have H, then we have H, OH, and this is H. Okay, this is known as what? Remaining we have CH we have. Then you can see that here there are OH and O group will be there, that we have to take in for the writing the structure of Hawa structure for the glucose. In the Hawa structure of the glucose, you can see that we have OH, then we have OH, then finally we have H. I think I have missed one more. So the structure will be like this. Look at the now. four OH bonds will be there, three OH bonds will be there. How to write? Pick up the right hand side of your glucose structure, OH, OH, H, and H will be down, then again OH, and this CH2OH will be not inserted inside this ring because it's a bulky group. We have to write it on upside. Okay, so this is known as what? Glucopyranose. Why it is known as pyranose? Because the entire pyranose ring will be like this. Okay, or you can write the structure like this also. The pyranose ring structure will be like this. In order to write it in a similar way, we are writing the structure of glucose by comparing with the pyranose. Hence, it is known as alpha D plus glucopyranose and beta D glucose pyranose. So, reverse of this, you are going to write for the alpha structure of the alpha form as well as with the beta form. Next, write the Hava structure of maltose. Remember, in the maltose again, it is nothing but your 6-membered carbon chain, okay, consists of OH and a H as well as with the CH2O group. First, you write the alpha form of the maltose, then with the beta form of the maltose, then connect the carbon 4 with the oxygen bond. Okay, so that is nothing but your glycosidic linkage. So, and shows that this is a Hava structure. Next, what are essential amino acid? Give an example. So, this is a 2-mark question. Remember, we know that few amino acids can also synthesize inside our body. They are non-essential amino acid. Remember, if the amino acids are synthesized in our body, it's not necessarily taken through the diet. Such amino acids are called as non-essential amino acid. But few essential amino acids we have to taken through continuously through the diet in order to provide a amino acid content to our body. Such amino acids are valine and leucine, and it should be continuously taken through the diet. Okay, it is required by the human body, but it cannot be synthesized by our body, but it can be taken through the food. Whereas the non-essential amino acid, which is required by the human body and it can be synthesized by the human body, example glycine and alanine. Next, write the differences between the fibrous and a globular protein. So, as I said, proteins are the polymers of alpha amino acids. There are two types of protein will be there, fibrous proteins and globular protein. In fibrous protein, you know, the peptide linkages are parallel to one another and they have a hydrogen linkage and a carbon disulfide linkage. And it looks like a threads, hence it is known as what? Fibrous protein. Example, the keratin and a myosin which is found in the human body. Keratin, we can take an example of the hair and the cell, and for the myosin, we can find it in the muscle. Okay, coming into the globular protein, you know, the peptide linkages of the amino acids, you know, they called one another and forms a spherical shape, which we call it as what? Globular protein. Example is insulin and albumin, which is found in the human body. This type of proteins are usually soluble in water, whereas fibrous proteins are insoluble in water. Next, give an example for each type of hormones. Polypeptide hormone, amino acid derivatives and steroid hormone. Okay, so we know that polypeptide hormones are of two types, insulin as well as with the glucagon. So you can write with the example of insulin and a glucagon. Insulin always controls the blood sugar level, whereas the glucagon, you know, whenever the glucose level is required for our body, this glucagon will break the carbohydrate and provide the glucose. Okay, which increases the glucose rate. Next, amino acid derivatives, we can expect two types, that is thyroxine as well as with the epinephrine. Thyroxine, which are produced from the thyroid gland, which maintains the normal growth and development in a human body, whereas epinephrine will be controls the blood pressure. Next, what are steroid hormone? Steroid hormones are of two types. One is glucocortical hormone and the one is sex hormone. Okay, so in the glucocortical hormone, again we have two types, glucose corticol and mineral corticol. And in the sex hormone, you can see the three types. One is testosterone, estrogen as well as with the progesterone. Okay, testosterone, estradiol, as well as with the progesterone. Testosterone are produced from the testes of the male and it produces secondary sexual characters in the male, whereas estrogen, it is produced from the ovary of the female and it produces secondary sexual characters in the female, whereas progesterone is also produced in the female and it is produced from corpus luteum, and this hormone, which are mainly responsible for preparing the uterus for the implementation of fertilized egg. Okay. Next, write a function of following hormone. As I said, insulin will be maintain the blood sugar level, thyroxine will maintain the normal growth and development, estrogen and androgen. So, estrogen and androgens are both the male and the female sex organs which are develops as secondary sexual characters in male as well as in the female. Next, define denaturation of protein. Give an example. So, we all know that what is mean by denaturation of protein, because we can see that denaturation of protein in our daily life also. Example, coagulation of egg white or curdling of milk is the best example for denaturation of protein. Then what is it? Which means that every protein in a biological system will have their own unique three-dimensional structure along with the activity. Suppose when we apply the stress in the form of increasing the temperature or by changing the pH, the protein can lose its activity as well as its structure. That process which we call it as what? Denaturation of protein. Example, coagulation of egg white as well as with the curdling of milk. Next, what are the biological importance of nucleic acid? As I said, nucleic acids are the biochemical material, which transfers most of the characters from one generation to another generation, hence which we call it as what? Nucleic acid as a hereditary material too. What are its uses? Let us see now. So, DNA is the chemical basis of hereditary and regarded as reserves of genetic information. DNA molecule is capable of undergoing some self-duplication during the cell division and forms identical DNA strands and transport to the daughter cell. And the synthesizes of protein inside the cell. Actually, the proteins are synthesized by RNA molecule in the cell, but the message of the synthesis of a particular protein was given by DNA. Okay, or present in DNA. These are the functions of DNA. So, this is all about the important questions which has been raised many times from the biomolecule unit. Remember, those who are taken the PCMB, it is easy for them to study the biomolecules. The same thing they have studied in the biology also. But for the PCMC students, it's quite difficult. But it is all basics that we have learned from your high school. So, it's just an extended part of it. Just try to understand the types as well as with the how to write the structure along with their uses, especially with the hormones and the vitamins, they can ask with the many types of question. And in this unit, as I said, vitamins can also be asked. Vitamins are of two types, water soluble and water insoluble, and uses of the vitamins and deficiency of the vitamins can also be asked. Remember, the water soluble vitamin as well as with the fat soluble vitamins can be asked. Fat soluble vitamins are A, D, E, K. Okay, whereas water soluble vitamins are vitamin C and vitamin B complexes. Any of this can be asked for 1 mark to write with a proper example. Okay, hope you all understood with this unit. I'll come back with the next unit. Till that, take care and be safe. Thank you.